Okay, so after that, we're making her sushi. No.
Do we like this shirt? I just got it.
Yeah.
Oh, you're like, what? We're from Target.
You're gonna leave sugar.
I got this new shirt. I got it from Target.
Free. Yeah, when's that? What?
I don't think she has it on like all day.
I mean, we made it now. We got 75. Where's that one? What time does the acclimate start? I'm probably gonna go to there now. 6? Couple of in my lane.
We're 10.
Oh, because all the fucking teams that we played are playoff teams. I know. So that's why our freaking rankings. Except for Alton.
Okay.
We got to do some more. Yeah.
You just do it Sunday. I'm gonna fold it.
But are you gonna go with Josh?
Yeah, and yet she's gonna play the rest of the semester because they gave us shutouts and showers.
It pissed me off when I do something.
What could you make of you guys? Like, my jaw was about to drop.
The judges.
Oh, it's Valentine's Day.
Like, told them they were too like this.
Wait, we can do it on birthday? Guys, we can do it Thursday. It's usually every other Thursday anyway, it's not Thursday. That's how they put it. Okay. That's why I said that. Like I can do it. It don't matter to me. That could be a girl that
Stifle, I always think of both.
I really don't care. Thursdays or no, let's just
Are you guys not on that? A big chart. I remember last week, it was just like before the game. And then it just
All right, guys. Let's start. Let's get started. Half the class is missing today. All right. I'll take attendance. Brianna Akins. Here. Drew. Here. Rhea. Here. Anna. Here. Nick. Here. Abby. Here. Hannah. Here. Rosa. Here. Matthew. Here. Jillian. Here. Mia. Here. Annie. Brianna. Hills. Okay. Brandon. Here. Katie. Jessica. Here. Jesse. Here. William. Marissa. Anna. Matt. Sophia. Aubrey. Here. Matthias. Here. Colin. Here. Olivia. Here. Braden. Amber. Here. Katie. Here. Ava?
Here.
Kendall?
Here.
Emily?
Here.
Lauren?
Here.
Jen?
Here.
Isabel?
Here.
All right. Okay. So today is an exam review day. Reminder, your first exam is on Wednesday. It'll be in this class. You'll have the full 50 minutes to take it. It'll be 50 multiple choice questions. And so, I'll be here if you have any questions. The material on the exam, anything from the lectures is fair game. The book is great for really understanding and comprehending those principles, but if something is in the book and it's not in my lectures, then it won't be on the exam. Any questions about that? Please bring a number 2 pencil or just a regular pencil because it will be taken on a Scantron. Is anybody not familiar with Scantron use? You guys all used Scantrons before? Okay, so it will be on a Scantron, and so I'll go over the instructions before we begin on Wednesday. I think that's all for announcements. If you weren't here on Friday and you did not send me the second worksheet assignment, send me that via email so I can give you points back. And today I just have all 4 lectures pulled up. I'll kind of open the floor for you guys. Did you guys see the study guide on there? Was that helpful? All right, so yeah, we can just go through whatever questions you have on the study guide or just from the material, and then, yeah, I'll answer any questions you have. Reiterate any concepts you still don't understand. This is a really good time to ask those questions. Yeah.
Can you go over the ABC transporter system?
Yeah. So the ABC transporter system is a type of active transport, so it requires energy. That energy is in the form of ATP. It's a really large family of a transport system. So there's 2 types. There are ABC transporters that are able to take nutrients in, and there are some that are used to pump things out. So some bacteria, if you're trying to target them with antibiotics, they start to learn and adapt that you're trying to kill them with antibiotics. So they have these ABC transporters to kind of pump the antibiotic out so that it doesn't harm them. Others will use it in a way to take in critical nutrients that they need to grow. So here the example that's shown. So you have a bacteria. This is the bacterial cell membrane. You have, so right here you have a cell membrane and then an outer membrane. So I guess, is this a gram-negative or gram-positive bacteria? You have the 2 membranes. It's the 2 membranes, not the 1. It's going to be gram-negative because gram-positive cells have just this inner cell membrane and then a really thick peptidoglycan layer. Gram-negative cells have a thin peptidoglycan layer, which is not shown here, but they have this outer membrane as well. So because of the ability of gram-negative cells to have these 2 outer membranes, they have this system where you can have 1 protein transporter in one membrane and 1 in the other membrane. They utilize both of those membranes. So over here you have this red circle. This is a solute, something that is needed to get into the cell. And the solute essentially will need to get into the cytoplasm, so the inside part of the cell. So it's got to go through 2 membranes to do that because it's a gram-negative cell. So here we have this first one called a porin. It's essentially just something that allows things, it creates this pore in between the membrane, it allows the solute to enter through. So after the solute enters through, it's got to go through the other membrane. But in these ABC transporters, in this thing, this is the ABC transporter in green. In order for the solute to be able to attach here, it's got to be brought in by this other molecule called a solute binding protein. It's just a protein that binds the solute. It brings it to the ABC transporter, and when it eventually touches the transporter, there is ATP hydrolysis that occurs. So it's driven by energy, ATP, it's active transport. It drives this conformational change, and then it allows this transporter to open up on the other side of the cytoplasm, and it can then release the solute to the cytoplasm. So the goal here is to bring in a molecule, a nutrient. This is the example of uptake. It's bringing in this molecule, but there's two membranes to go through. These are not permeable to solutes on the outside, so they need help to get in. Does that help? Does that make sense?
So if it's the other way, then it would just be reversed.
It would be exactly the same, the other way, just reverse. So if it wants to take something, if something's in the cytoplasm, you know, if we're giving it, if we're trying to target the bacteria with an antibiotic, you know, and the antibiotic is in the cytoplasm and it wants to pump it out, then it would just go in the other direction. So the reason it's called an ATP binding cassette is because essentially when you have that binding, the ATP hydrolysis occurs, and so it causes the conformational change, which is a recurring theme that we'll see a lot in microbiology, this conformational change of proteins. So this is like the in-depth explanation of how this works. For the exam, it's important to understand how things work, because then it'll help you for the exam. But for the exam, you know, you should understand, you know, what type of transport it is, you know, what, if it's like what it can be used for, uptake and efflux, and, you know, is it driven by energy? What's the energy type? That kind of thing. This is utilized by a lot of gram-negative bacteria because of these two membranes. What else?
Could you go over like the difference between gram-positive and gram-negative cells again?
Yes. Okay. So, gram-positive, can you guys see this picture okay? All right, so this is chapter 3. So gram-positive cells, all cells are going to have a cell membrane, right? The phospholipid bilayer. You have the fatty acid tails that are hydrophobic, which means that they don't like water and things, they don't allow things in, and then you have the parts facing outward, which are the phospholipid heads. And so, you know, both are going to have this membrane. Gram-positive cells have this really thick peptidoglycan layer, and gram-negative cells have a thin peptidoglycan layer. But because of this, they have this outer membrane over here. So it's just another phospholipid bilayer. Now remember, these membranes have these embedded proteins and porins and things to help things go in and out. So that's why you'll always see these porins and these membrane proteins inside of these membranes. Gram-negative cells, because they don't have a really thick layer over here, you know, there's got to be something connecting it to this outer membrane. So there are these lipoproteins that are just connections. They're literally proteins that also have some fats on them, and so they connect the membrane to the peptidoglycan layer. You don't see that here. The top part of the gram-positive and gram-negative cells, they have this S layer, which is essentially just this, how did I describe it here? It's an additional protective layer. So it's a protein, and so the cell membrane really helps to confer the shape of the cell. But you have this additional protective layer that just kind of helps to make sure that it's, you know, it has just another layer. It's just like another layer to make sure that, of protein, that to make sure that the cell is not going to be exposed to osmotic stress and just other stressors in the environment. In gram-negative cells, in between the S layer and the outer membrane, you have this LPS, this lipopolysaccharide, which are literally in the name. They're just this outward leaflet and these porins that are attached to the outer membrane. So there's also lipoproteins. So these are polysaccharides, so these are multiple of them in chains, and so these are just the proteins. So those are some differences. But in gram-positive cells, you don't have those extra lipopolysaccharides or lipoproteins because you have this really thick peptidoglycan layer. These are just some sugar chains that are outside of the cell. Kind of think about this. This is the S layer around it, protecting it. These are just some sugars sitting on the outside of the cell. But one key thing that I want you guys to know about gram-positive cells is that this peptidoglycan layer has these teichoic acids, these acids that essentially help link all the different levels of the peptidoglycan layer. And does anybody remember what this is important in? Yeah. Exactly, yeah. So when we're doing a gram stain, the iodine actually interacts with these teichoic acids and the crystal violet to lock in the crystal violet in the gram-positive cells. Does that make sense to everyone? Does that help answer your question? Yeah, so this capsule is essentially what these glycosyl chains are made of. It's just another added layer of protection as well. Yeah.
So I guess again, my question is like in the gram-positive cells, like the layer of peptidoglycan is really thin, but do they still have these acids within them?
In the gram-negative?
In the gram-positive, I mean, yeah, gram-negative, I meant, yeah.
Yeah, they don't have the teichoic acids because it's such a thin layer, so the teichoic acids kind of help to link them all together. So if there isn't as many of them, they're not going to be, there aren't the teichoic acids, yeah.
The iodine interacts.
The iodine is what's interacting. It interacts essentially with the crystal violet stain and the teichoic acids to lock in the crystal violet. That's why it becomes irreversible and the gram-positive locks in there, or the crystal violet locks in there. Whereas in the gram-negative cells, they don't have those teichoic acids, so the iodine doesn't interact with them and lock in the crystal violet, so the crystal violet becomes reversible. You can remove it. And what's the step that removes the crystal violet from gram-negative cells? What's the, which part of the stain? Ethanol. It's the ethanol. So the ethanol is the decolorizer. It essentially removes the color. So that is, so in a gram stain, it's a differential stain. So we sometimes often call this decolorizing step with the ethanol, we call that the differential part of the stain. That's the differential stain, because that's the part that allows you to differentiate between gram-positive and gram-negative cells, because that's the part that removes the color from the gram-negative cells, but it doesn't do anything to gram-positive cells because it's already been locked in there. Just a reminder, the gram stain. Procedure, you do crystal violet. Then you do iodine. You do ethanol with washing with water in between, and then saffron. Ethanol is the differential step, aka decolorizer. The iodine, say it complexes with nucleic acids, lock in crystal violet. This is in gram-positive cells. They have nucleic acids. Does that make sense? Should know the order of these as well.
So could you call the last step like the reverse sort or like
The saffronin?
Yeah.
The saffron, that's a good question. It's actually called the counter stain. So that's exactly like along the same lines of what you're trying to say. So this is the counter stain. It's literally the other stain.
And this is kind of like what strips away the purple if the purple can be stripped away.
No, the ethanol strips away the purple. The saffronin is just the stain you put on top. And because the gram-negative cells are going to be clear at that point because you put ethanol, they are able to stain the purple, or the pink, the saffronin. The gram-positive cells have that purple locked in, so when you put a counter stain on top of it, it's lighter than the crystal violet, so it's just not going to overpower it. It's not going to overshadow it. Yep, so the two stains essentially are the crystal violet and saffronin. Those are the two stains. This is the initial stain, this is the counter stain. But these two steps here, the iodine interacts with the actual peptidoglycan layer, the cell wall teichoic acid. And so because they're different in their cell walls, this is a very important step. The ethanol is also very important because if this one is done right, then this one will allow for the differentiation where at this point, gram-positives will be purple, gram-negatives will be clear because it's a decolorizer. Yeah.
You see gram-negative is purple?
No.
Off of the ethanol stage?
Before the ethanol stage, the gram-negatives are purple. When you put the ethanol on there, the ethanol washes out the purple, and then when you put the counter stain, the saffronin on top, then that actually sticks. So let's go to, does anyone remember where, was it chapter 2? Stain, and then we have the gram stain. So before you do anything, when you heat fix the cells, you're going to have just clear cells. We haven't added any stain. Then you add the crystal violet, and basically the crystal violet is going to be taken up by both peptidoglycan layers. But look at how it says here that it's still reversible. They stain the cells reversibly. So then when you add the iodine, it binds the stain to the gram-positive cells. So now iodine complexes with crystal violet, which allows the stain to be retained. Whereas in gram-negative cells, the iodine is not complexing with the actual crystal violet, so it remains reversible. Then you wash with ethanol, and because this one is now permanent in there, there's no change, but now the stain in gram-negative cells is reversible, so you wash with ethanol. There's the decolorizing step, which is also the differential step, because at this point you can differentiate between the two. Then you add the safranin, it's the counter stain. It will counter stain gram-negative cells, but it won't, it will absorb the safranin, but they will remain the dark purple. So if I were to ask you, what if we washed with ethanol before putting iodine? What would happen? If we had flipped the iodine and ethanol steps, if we had done crystal violet and then ethanol, what would the gram-positive and gram-negative cells look like? Yeah.
But they just the iodine like interacts with the acids like hold the crystal violet in, so that the crystal violet kind of like not be able to like seep in or lock in.
So if we went through the entire step, yes, they both would be pink. But I'm just saying stop at that step. So we do crystal violet and then ethanol. What would they look like? Yeah, they're all going to be clear. But you're right. If we were to go through with the entire thing, then we haven't locked in the crystal violet, so they would both be pink if we went through the entire procedure, if we had flipped the two. But at that step, the crystal violet would actually wash out. So the iodine is really important for that. Does that make sense? Okay. What else? Yeah. Yeah. So, for a spore stain, I don't really need you guys to know a lot of the details because we will talk more about spores and we will actually do this staining with spores. But just, one of the important things is, some bacteria, some fungi, they make spores. These are structures that allow them to kind of hibernate, hunker down, and just go quiescent. And then when they need to grow and, reproduce, then these spores kind of pop out and allow for that to happen. So we can sometimes stain cells and stain their spores at different colors. So here we see the spores are in this malachite green. And so you can see if a cell makes spores, you can use a spore stain to be able to identify, the spores from the cells. A negative stain is essentially, instead of coloring the actual cells, you're coloring the background. So it helps to make things like the capsule of bacteria more visible. So the capsule is, the negative stain, meaning it's whited out because the background is stained instead.
Not as like in depth of a question, but for the chapter 3 bullet points, one of them is asking about like what structure defines the existence of a bacterial cell, and that's the cell. And then it asks what is it composed of, but then another question you asked what are cell walls made up of?
Yeah, so it was actually in the title. Cell walls are made of the peptidoglycan layer essentially, but there's obviously more to it that has to do with the gram-positive, gram-negative differential, just the differences in them. And then the step where I asked, what was the first part?
What is the structure that defines the existence of a cell?
Yeah, so that's the cell membrane. That's the entire membrane. The structure that defines the existence of the cell is the cell membrane. The cell membrane is not the cell wall, it's essentially the membranes in between. So remember, gram-positives have one membrane, gram-negatives have the two membranes. So the cell membrane, and what this really means is, if you think about it, a cell will have, you know, the cytoplasm, a bacterial cell has the cytoplasm and all of that, you know, all of where the nucleoid is in and all the DNA. And so that's essentially going to be a bag of soup if you don't actually have something to confine it in. So the cell membrane just helps to confine the contents of the bacterial cell. So that's what I mean by it defines the existence of a cell. If there's no cell membrane, then you don't really have a cell.
So the difference is that the cell membrane is made up of like phospholipids.
The phospholipid bilayer, yep, and then you have proteins that are embedded within, on top, around, so that's why half of the cell membrane is essentially protein, because you can't really forget about the proteins that are embedded in it, even though the cell membrane, you know, the phospholipid bilayer is the phospholipids, the phosphate heads and the lipid tails. Remember, there are, you know, the cell membrane, it helps define the existence of the cell in terms of, you know, making sure it has this confined structure and support. But there's other things at play that kind of help make sure that things are still stiff and it's, you know, not just completely falling apart. And so there are things like hopanoids, these stiffening agents. So it's important for you guys to get used to, you know, you see a cell membrane, there's the outside, which is what we call the extracellular environment, and the inside of the cell is the cytoplasm. What else? Yeah.
Could you review how microbial diversity implicates genetic diversity?
Yeah. Okay. So microbial diversity just means that, you know, we have a lot of different diverse microbes. And some of those differences really occur in their growth rates, where they can live and survive. There's just so much diversity in those things. And there's so many specific niches for microbes, so they can adapt to different temperatures, really extreme temperatures. They can adapt to different pHs, salt environments. There's so many physical things in the environment that they can adapt to. So why does that matter? Well, if microbes are able to do all these different things, then that means that they have genes that are able to allow them to exist in really, really cold or really, really hot temperatures and really different pHs. So that microbial diversity really means that there's a lot of diversity within their genes. And remember, because they're able to adapt so much to the environment, they have this ability to have this genetic plasticity, so they can turn genes on and off as they need them. So if we have this diversity in the genes, well, what do genes make? Remember the central dogma of life. We have genes. Our genes are encoded in our DNA. I shouldn't say our because we're talking in the context of bacteria, but the genes are encoded in the DNA of the bacteria. The DNA goes through transcription and becomes RNA, messenger RNA, and then the messenger RNA is translated into proteins. So genetic diversity eventually means that you have protein diversity. They're making proteins that are allowing them to survive and withstand all these different environments. Now, what do proteins do? Proteins have a function, and proteins are enzymes, so they allow for different biochemical reactions to occur to allow them to survive those different environments. And so because there's enzymatic diversity, then you have all of this biological function diversity. Does that make sense? What else?
Can we go over the archaea a little bit more?
Yeah. Okay. So archaea were not really well defined for a long time. Scientists thought that they were just these unusually looking bacteria. But over time, as they started to develop more microscopy techniques and techniques to see these microbes, they started to realize that, yeah, maybe they look more like bacteria, but they actually have some eukaryotic features as well. Remember, bacteria are prokaryotes. So, there are things that we started to learn that make archaea actually a little bit more different from bacteria than we thought. And some of those things are their cell walls. Archaea also have cell walls like bacteria, but their cell walls are a little bit different in their composition. You don't have to remember the composition of each. They also have cell membranes. Remember, cell walls and cell membranes are different. So those are chemically distinct. And then obviously genes, they have genes as well. And so archaea actually have more similar gene expression machinery to eukaryotes than to bacteria. So they're understudied. We thought that they were bacteria for a long time, but then as we learned more, we saw that they actually maybe resemble eukaryotes a little bit more than bacteria. And so these are some of the ways that they differ from bacteria. They're still pretty understudied. So the other thing, another thing about archaea that you guys should know is that they are also, a lot of them are extremophiles. So they're similar to bacteria in the way that, how we talked about all this bacterial genetic diversity, being able to adapt and survive in these crazy, environments. Archaea also are able to do that. They can live in all these really extreme temperatures, pHs, salt environments. Yeah.
Can you go over the differences in fatty acids and phospholipids?
Do you have a specific slide? I assume it's not this.
It's in chapter 3.
Fatty acids. Do you have a slide number? Sorry, I'm having trouble remembering where exactly it is. A little bit. Okay, so do you mean part of the cell membrane, the fatty acids and phospholipids that make up the cell membrane? Okay, so yeah, the phospholipid membrane, it might be a little bit easier to draw this one out. So all bacteria, say this is a bacterial cell, you have the nucleoid, the DNA, you have other proteins and things like that. So this is the cytoplasm of a bacterial cell. So this is just a bacterial cell. Now if we zoom in, there's the cell membrane, and then there's also the cell wall. But we need the cell membrane to essentially confine everything in the cytoplasm. So if we zoom in on just what the cell membrane is made up of, it's made up of phospholipids. So these heads, and then these tails. So if you zoom in, so now this is essentially the outside. So this is like this part of the cell. And then you have this right here is the cytoplasm. These phospholipids, essentially, these heads are polar, which means that they are able to interact with things, you know, in the cytoplasm and outside of the cell. They have charges on them, but the lipids inside, so this is a phospholipid bilayer because you have two layers of phosphate group heads and lipids, fatty acid lipids in the center. So these face inwards towards each other because these are nonpolar. And what that means is that they don't allow polar things to go in and out. Remember, things are always trying to go into the cell and out of the cell. So because these are nonpolar, it restricts things from coming into the cell, which is great. That's why we have membrane proteins, these proteins that allow transport into the cell and outside of the cell. But the difference is that these are polar heads. These are nonpolar tails, which are the fatty acids. And so this is the phosphate groups. And so essentially, you know, these are able to interact with things, but these are not. And that's why these are called hydrophobic and these are hydrophilic. It has to do with the polarity. Does that help a little bit? Good question. That's why we have porins, we have proteins, we have all these things embedded to ensure that things can go in when they need to go in and things go out when they need to go out. Yeah, binary fission. Do you remember the 3? Oh, was it in a different? Well, binary fission essentially is the way that bacteria can divide. It's their cell division. So, you know, we have to go through, I don't know if you guys have taken genetics yet, but, you know, we have to go through a completely different cycle of ways that we mate and that we make babies. But binary fission essentially is the bacteria cloning themselves. It's an exact copy or an exact clone, and because of that ability to essentially just clone itself, you have they're able to do it really quickly, and so that's one key feature of bacteria and why they've been around for so long, because they can divide and replicate so many times. And so when a cell, so you have one bacterial cell, and then you eventually want to have this divide into 2. So the first step has to be that all the DNA and everything in here has to replicate. So once that replicates, you have this version, and then you have another version of it in the cell. And then there's other processes that occur where now that the DNA has replicated, now the cell itself has to divide. So there's DNA replication that has to occur first, and then there's cell division, which is binary fission. So then you start to have this cell starts to divide into two, and so we go through meiosis and mitosis, but then eventually this structure that starts to form is the septum. And so this is essentially the step, or septation, is the step where you've essentially, this will pierce and you'll have two cells, or cleave and you'll have two cells. So septum formation is the step that allows you to see that there's going to be two cells. It's helping to complete cell division. You don't have to know the origin of replication, the termination site, and all of that for this class. That's all Bio 131. But you should understand, binary fission is cell division of bacteria. So a lot of times I actually used to get confused because I'm like, well, you're replicating it. You go from 1 to 2. So why is it division? Well, yeah, you have DNA replication at first, but then you eventually have to have the cell divide into 2. So division is in a way replication, but there are differences there. Does that help? Does that make sense? Okay. What else? Yeah.
Can you talk about why we target the double replicate or antibiotics?
Yeah, absolutely. So, bacteria have a cell wall, right? We don't have a cell wall. So antibiotics are used to treat bacterial infections in like humans and mammals. And so because mammals don't have a cell wall, we can target the cell wall of bacteria with antibiotics, because when we put the antibiotic, then if it's only hitting something that has a cell wall, then it won't harm us. It'll harm just the bacteria. So that's why we use peptidoglycan, because it's something that's interrupting cell wall formation. We don't have cell walls, so we're not going to get hurt. What else? A few more minutes. Ask any questions now or forever hold your peace. I'm kidding. You can email me or come to my office. All right, good luck studying. If I'm not in my office, find me in the lab. I'm actually gonna try to create some lab around too, just to start working on stuff, so we can go through that too.
Carboxyls are structures in cells that essentially allow for sequestering or like keeping in ribose codes and enzymes. But essentially what you need to know is that carboxyls are this organelle, quote unquote, in bacteria, because remember bacteria don't really have organelles, but there are some organelles in some bacteria. There are some that have these carboxyls, and they help like just essentially doing photosynthesis and keeping and producing carbon dioxide and things like that.
Yeah, okay.